Good morning, everybody. I'm afraid that organizer has proposed to me to be the chairman for decision because Mauricio is not going to come. He has several family issues and she regrets absolutely not being able to be here today. So it's my first time doing chairman, so please excuse my absolutely incompetences and inexperience in presenting people and managing the times. In fact, I have been waiting 5 minutes because they specifically told me at 9:30. So I am the first lecturer today, and let me introduce them. I'm going to talk about the recent advance in comparative pathology for one of my specializations fields, Rift Valley fever. I am veterinary pathologist, so I am in the buccal net from different fields and diseases, but it's one of my major topics. Rift Valley fever virus is a Bongaviridae single-strain RNA virus, quite familiar with this family. It has a pan-haloid structure very characteristic of Bungaviral family, and one of the most tricky things about this genus of flavivirus is they use coupled primers of the host cells to transcription and to replication. This is the virus for which I'm talking about. It's composed by 3 segments: large, medium, and small, and all of them are codifying through the coupled primer a RNA dependent ribopolymerase. The M segment is divided in 3 different proteins. It's coding 3 different proteins. The 2 principals, GN and GC, they are structural. They belong to the viral envelope, and they mix with different components of cell membranes of the host cells to conform the bilateral capsid. And the other one are non-structural and divided in 2. The one in which is fused with the GN, or one bigger one of 78 kilodaltons, named NSM2. That is, all of them are involved in different steps of the transcription and the replication and so on. The double ambisense segment, S segment, is quite characteristic of flavivirus genus, and in the regular sense, they produce the nuclear protein, clearly involved in the process of replication, and in the ambisense, the protein translated is the NSS, very quite important in avoiding immune response systems. Here is, in fact, a quite fast review about the major function doing by the viral proteins codified in the virus, and these are the target of all vaccines developed until now have been designed for giving answer against these viral proteins. So we have actually 3 viral living attenuated vaccines: the Smithford vaccines, MP12 and Clone 13. There are a lot of regulatory concerns because they are not safety. They provoke abortions, they provoke high-pitched fever, they provoke disease at the end. So we are working quite, oh, it doesn't work. Okay. We are working on other types of vaccines, most of them focused in the ENS-M, ENS-M2, and the ENS-SS, in the recombinant ones using alpha virus, Newcastle disease virus, different platforms of adenovirus, even different vaccinia, modified Ankara vaccinia virus to vector. And there are several attempts to do DNA with quite successful in the case of GNGC. In fact, there are 3 human new vaccines against Rift Valley fever virus based on DNA, different platforms. They are in between phase 1 and 3 in the regulatory process, and just one that is undergoing under regulation today is in preclinical study belonging to the group of N. we all scientists, we are focused on the pathogenic mechanism involved with the NSS and with the GCGN, because they are major antigen. They are clearly related with the creation of a strong antibody response. They modulate apoptosis and, more important, inhibits host transcription and provokes cell cycle arrest. They are quite related with pathology that we observe with this virus. In fact, the NSS has been widely described in the literature in mouse and different models that I'm going to discuss later, because of they have all these kind of actions leading to these consequences. They clearly stimulate apoptosis, necrosis, and a lot of different disbalance between pro-inflammatory and pro-apoptotic cytokines. At least for us, the most important actions related with pathogenesis and related with pathology observed in this disease are these three one. First one, blocking type 1 interferon production and disruption of whole set transcription are the leading one. But not less important is the binding to pericentrosomatic DNA repeats that it's mediated with NSS plus the RNA-dependent ribopolymerase. What does it lead? It leads a chromatin deregulation who stimulate and who promote and who provoke the apoptosis. The SNAP30 cause a repression of interferon beta transcription, maintaining this repressor complex. And nowadays it's thinking that this regulation between the type 2 interferons are Th2 cell lyset is leading to an immune evasion, an immune system evasion of the virus, leading to the failure of most of the vaccines that we are developing now. Host susceptibility. Domestic ruminants, wild ruminants, rodents, carnivores, non-human primates, and of course, humans. What is the most tricky situation when we design modelization, animal modelization against this virus? There are clear age-dependent affectation, susceptibility. However, most of our more used models for regulatory purpose are not useful for this disease. So pigs, dogs, guinea pigs, they usually don't have consistent viremia. They don't usually have consistent symptomatology. Even reactogenicity is not very clear. But however, it's incredible, but the most susceptible species, calf, sheep, and for example, non-human primates, abarks less than 35% of papers done in this illness. So it's quite bizarre because this illness was firstly described in the beginning of the last century. Human disease. We want to mimic human disease, and it's characterized by 5 syndromes. The most common by far is self-limiting febrile illness. It affects 7% of the people infected. Due to the two biphasic and covalent phases, they go through all this flu-like syndrome, go to a recovery, or due to a second mechanism that is quite long and is not the topic of this talk, they go to a vision loss or neurological disorders. The hemorrhagic fever and thrombosis are residual that affect less than 50% of human patients, but however, vision loss becomes to blindness in at least between 10 and 15% of Rift Valley fever affect human patients, with the dramatic consideration for their quality life. Neurological disorder usually are related with another types of syndromes involved within the patients, but however, they don't seem to be very related with the rest of the syndromes. We have several models running now Mouse, for sure, they are susceptible. They usually die between the 2 weeks, corresponding with the human disease. They are divided in two major syndromes, an acute necrotizing hepatitis and a later stage with a meningoencephalitis. They are cost-effective and they have been widely used for transgenics, for pathogenesis, so on. What's going with them? The most important disease they are involved in after the self-limiting febrile phase is not diagnosed here. No hemorrhagic fever, but in spite of all, it's a quite good model. What happened? We can see here which are the widely in immunohistochemical change staining at 2, 3, and 6 days post-infection, leading to the curve that is suspected to be there. So that corresponds with the human situation. We can see as well different parts of apoptosis and hepatocytes. Here you had a microscope on electronic who leads, this is the normal situation, and you can see karyolysis, karyorexis with the disgregation of the organella and so on, even leading to a mononuclear inflammatory foci that are surrounding all the hepatocytes affected. Even we can see here, maybe you have to very good very good at looking. If you can see as me, please believe me, there are intranuclear inclusion body inside the hepatocytes in every single stage, no age dependent. That's a clear point of this mouse. And in a late stage corresponding with the beginning of the meningoencephalitis, we start to observe hepatic regeneration, just the same in human beings. Here is the affectation in the latest stage, in which we hear an chiasma optic, in which we can see the tubercles and the rapha. It's quite important because the mouse start to tremble. So it's quite impressive. 1 hour later, to start with this symptom, the mouse died. So it's quite fast, the spreading of the virus into the nervous system. We see the non-suppurative lymphoplasmatic meningoencephalitis with a clear staining in olfactory mucosa. Mouse has been challenged by every single route that you can imagine, intraperitoneal, intracranial, aerosol, all that works, all that cause pathology, but however, only the aerosol ones lead this stain. Rat. Rat is quite similar to mice. Even one report in which I have several concerns about the diagnosis used suggests the presence of uveitis, but it's good because Wister Fisher, brown Norway rats are quite susceptible, but however there is strong variability between breed strains. In fact, 121 or even Charles River inbreed strains between Europe and North America have different susceptibility to Rift Valley fever. That means that there is a strong genetic influence in this model for the behavior and the susceptibility in pathology of this mouse, leading to a not very well, not so consistent that we wanted. Of course, there are the age-dependent differences as in mouse. So I like because it's easy to manage, it's easy to breed, it's easy to work, but there are sort of disadvantages that I prefer another type of models. Hamster, definitely the point with this model is they're not resources. There's not antibody, there's not enough reagents. There are a lot of problems involved, and the most tricky situation with hamster that I don't know if you're confident working with this, but I don't want to be bitten anymore for a female hamster because they are extremely aggressive. So if you use this model with a neurological affectation, they're becoming crazy and they're going to kill you and your technicians. So maybe it's not a good idea to manage hamster with Rift Valley fever virus. Herbil, less than 1% of papers written is working with Herbil, but however, it's quite good for neuroinvasiveness because female hamsters are quite aggressive, Herbil's not. Herbals are softly, even they lick you, so it's incredible to work with them. Disadvantages, no significant diseases, they don't mobilize anything else than encephalitis, so they're not good for safety concerns, they are not good for vaccinology. Sheep, adult IWI, they develop hemorrhagic fever, they develop a strong ocular pathology, but the problem, high rate of abortion and fetal malformation. Do you remember that I told you in the beginning of the presentation that one of the most tricky events of the NSS is that they have a strong effect in the chromatin deregulation, host cell transcription, cell cycle arrest. They have this clear effect in the placenta, in the dicotyledonian placenta of IWIS, leading to a malformation and leading to a late abortion in most of the 70%, 75% of the affected IWIS, pregnant IWIS, so it's quite a lot. For a comparison, human pregnants abort in 5–10%. It's believed that is caused by the interleukin 6, no by the Rift Valley fever infection. It's the fever who leads them to abort, not the virus. However, doesn't work anyway. They had a breed-dependent susceptibility to isolates, and of course, it's quite expensive to work with sheep. Lambs are quite susceptible. Later acute hepatitis. Hepatitis, to say something, because it's more necrosis, it's more hepatic necrosis more than necrotizing hepatitis. So it's quite fulminant, but they are useful to detect the effect of colostrum from vaccinated IWIS. That is quite useful in the endemic areas in Egypt, Arabian Peninsula, so on. However, they are not affected by neurovirulence. Normal, they don't have any time. They die before to become the meningoencephalitis. And they don't have any ocular diseases. However, sheep and goats are the best models today. These are the pathological signs that you can see: extensive hemorrhages from spleen, limbs, rumen and digestive pattern. The blue red eyes, so famous in the human beings, they became blindness in 15 days. And the necrotizing hepatitis with a arthrogryposis in 2 aborted fetus. This belongs to the draft and Oda sheep breed. There are another types of breeds. They are affected in this manner in the southern part of Africa, South Africa and the Horn of Africa, that the problem is more neurologic than arthrogryposis, but who knows. Non-human primates. Non-human primates develop, almost always they are affected by lethal hemorrhagic fever. In fact, they mimic less than 50% of human syndrome. So it's similar clinics to humans, but only in a syndrome that is not quite common. It's quite important for testing safety and vaccines because for regulatory process, but they had a species breed dependent pathology quite sharp because of they have different, they are, how's it said, African monkeys, New World monkeys, old monkeys, the behavior are absolutely different in pathology. For example, marmosets are quite affected by them, baboons not so many, and rhesus macaques only by fever. In just one case has been reported with the lesions that I'm going to show you before. So I'm going to show you after, but it's not so common. For sure, you need to keep monkeys, and it's quite expensive to work with them. This is a marmoset, common marmoset, who develops the typical hemorrhagic fever with nerval infarcts. It's in the cortex, in the cranial cortex, near of the chiasma optic. Here you see the thrombus, and here is the neural infarct. Thrombus in lungs, thrombus in kidneys, thrombus in choroid, in the external of the vessels, thrombus everywhere. So they die too fast, and with a not typical Rift Valley fever syndrome. This is the rhesus macaque that I talked about you. This is an African. He comes from the Africa directly. We were not sure if it was become so huge meningoencephalitis with the hemorrhages were clearly related with Rift Valley fever or something else was going on here. But the problem is that when we stained it, we clearly saw the neurons stained with Rift Valley fever, so it was clearly affected. He died for this. Other model, I'm talking about dogs, ferrets, guinea pigs, had the species-specific pathogenic studies, but the cost and the variability, at least for us, they are not worthy to work them. I'm going to present one model that we have developed in collaboration between our company, Warpasol, and CRESA in the Autonomous University of Barcelona, in which we develop in this breed of sheep, it's an European inbreed sheep, quite homogeneously, genetically speaking, this type of challenge model in which we do several shows with them, and most of them are study dependent, so I'm not going to go through this. But the main points that we use 60, 70 days lambs. They are in the frontier between lambing and iwin. So they have a lot of hormonal changes here. They become sort of resistant person, but not so resistant to not develop all the syndromes that you can see. We have tested a lot of different strains of Rift Valley fever virus from Egypt, from Kenya, South Africa, and we have tested at least 3 vaccines with this test. We have included in all of our experiments sentinel animals for natural transmission testing, quite important in the nature. What we see, it was a robust strain-independent Rift Valley fever virus replication with consistent viremia. Here you see 4 groups in which we measure in blood and in the in nasal settings what was the viral loads, and we observed no difference between all the isolated used. This isolating concrete was from mosquito, and the other ones were from cow and sheep, so clearly related with the species specific. We observed as well a variable shedding, but clear mimics the natural pathogenesis, because these mosquito ones, in spite of being in the oral several sort of variable setting, that corresponds in time, and that corresponds in percentage with you observing the nature. So in an affected breed, you observe less than 10% of IWIS setting, actively setting. So that corresponds with our observation in the model. The natural transmission by contact after subcutaneous challenge, they are our sentinels. Our sentinels become infected, delayed infected for sure, but they seroconvert, they start to shedding virus, and their behavior is exactly the same that the subcutaneous challenge. Again, is the only model that we have demonstrated this, not monkeys, not mouse, not other sheeps. Robust strains and environment-independent development of neutralizing antibodies with a clear correlation with GM and IgG dynamics. We measure. Yesterday I was I was wondering what Magdalena's talk, because we measure IgGs, total IgGs. I'm absolutely convinced and agree with you that we should go to divide the type of IgG, but that's what we do and that's what we work. Interferon induction was higher in cow and mosquito isolates, as expected. But the longer viremia, the lowest interferon induction. So that mimics the human situation, not the natural situation. And we thought that it's because there's something on this breed that makes it different. I'm going to stop because I would like to highlight the findings about the AUVEA, because as an immunoprivileged site, and there were not models developed until now to study it and to consider it in vaccinations, therapeutic and in the case of human, or prophylactic in the case of animals. Here we see an anterior uveitis with clear corneal edema, here we can see, and lymphoplasmacytic endothelitis, with clearly related with the vessels in the uvea, and starts going through the matrix. We see the blue light, the uveitis, unit to the endothelitis, and the most relevant one, a positive CD3 retinitis. It was our surprise that we couldn't see any mononuclear cell in the histopathology, but however, when we did the markers, we see a clear staining. So, and that corresponds with the nature, and that corresponds with the human situation, because humans are infected, at least 25% develops ocular sign, but only 9-10% become blindness. So we have mimicked the situation. This is our cases. And of course, we have the hepatic necrosis, we have the anterior uveitis, with the Rift Valley fever staining running in the lesions. And we got the lysozymes positive market, B cells in the hypopyon and in the mononuclear cells surrounding of the endothelitis and the uveitis, and most curiously, here in retina, surrounding the retinal vessels and inner in the paramacular layers, we observe, I'm sorry, I can see it very poor, but believe me, there are positive stains. So it's the first description in this retinitis in this modelization. Why? We don't know. We don't know why in this model yes and the other ones not. But the thing that it works, and that we think that it's that reproducible based models reproduce natural and experimental Rift Valley fiber infection in ovine. Such infection is strain and viremia independent. Anterior uveitis with lymphoplasmacytic endothelitis and CD3 positive retinitis have been described as a relevant condition in ribogesacib, mimicking the all-known human ocular condition. Immunological dynamics of neutralizing antibodies and interferon response in experimentally challenged sheeps mimics natural Rift Valley fever virus infection. And we think that ribogesacib is a robust, consistent, and suitable animal model for pathogenic, regulatory, and vaccination studies in this field. Thanks a lot for your attention. So, any questions? Yes.
Have you tried looking at MP12 or C13 live vaccines in your model?
Yeah, we tried to do it in Smith form because it was clearly related with safety concerns.
Yeah.
We obtained exactly the same results of the other colleagues. We obtained a slight bronchoplasmacytic subchronic hepatitis, non-necrotizing, so the animals can survive. And the dynamics of the infection, they can clear the virus, they have the Th2 bias, they got the interferon gamma increase, but the problem is that we are still creating pathology with, with this attenuation vaccine. So we are working on DNA and recombinant.
No, I didn't mean that. I mean, so actually the live vaccines are replicating what you see in animals in your model.
Yeah. And replicates again. Cowell, one colleague of us, described a very good, in another type of model with pregnant iris. He in fact got abortions, so it was quite surprising. Anyone? So thank you very much. Let's